Note that we did switch the order of the lectures. So today is neuropsychiatric disorders. And the newest syllabus is now the only syllabus on Blackboard. Go and look it up so you know what the readings are for the next 5 lectures. We only have 5 more lectures after this. Let's see. And then also, as I mentioned before, I'm going to have open kind of office hours between now and the last lecture of this class. I have most time Mondays after lecture, so if you have questions and you want to just ask me informally, plan to come up Monday after lecture. I have a shorter time Wednesday after lectures, but as always, you could always email me. I have more time Tuesday, Thursday, and Friday. So I invite you, particularly as you prepare for our comprehensive exam, note that the final is going to cover all topics. We'll focus on this subject matter that we haven't talked about yet, but I invite you to come and speak to me about anything that is confusing about the lectures or you want to clarify. So Mondays after class, starting today, we'll start today. I'll be available after class to answer any questions. Wednesday for a shorter time. And of course, anytime, just email me, wendy@cns.nyu.edu. It's on the information sheets that are posted on Blackboard. All right, so that is the announcements. So why don't we get started? So we are continuing today with the lecture we started last week on the study of emotion in the brain. And that study brought us to one specific structure called the amygdala. What we want to do is expand that and talk not specifically about the neurobiology of the amygdala, but more globally about what we know about neuropsychiatric disorders. And we're going to try and put this in the context of how we've studied different neuroscience concepts and brain systems throughout the whole part of, throughout the previous part of the class. So we started out in our lectures on emotion talking about why it's so important to talk about emotions. Emotions are the basis of many neuropsychiatric disorders. We happen to be able to be focusing on schizophrenia for this particular lecture, but I just want to emphasize that phobias, post-traumatic stress syndrome, panic disorders, all of these quite common neuropsychiatric disorders focus on fear. And you remember we talked about the major fear paradigm that has been studied, fear conditioning, where a tone is played at first, and then animals get paired between a sound and a shock. So tone and shock are paired together. And then after this, the animal is very scared of the tone, whereas before he was not scared of the tone. So just want to emphasize, we talked about several concepts or terminologies important for fear conditioning. One was the unconditioned stimulus, and one was the unconditioned response. Anybody remember what the unconditioned stimulus is? Yes, the shock. The unconditioned stimulus is that stimulus that you have a normal response to. Unconditioned response? What do you usually do to a shock? Yes. Fear response, so you actually freeze. There's lots of things that happen. The unconditioned response is the freezing that happens. This is the deer in the headlights response that the rat has. That also is associated with increase in heart rate, increase in blood pressure, and increase in stress hormones secreted in the rat. Those are all part of the unconditioned response. So unconditioned stimulus is the shock, and the unconditioned response is that normal physiological fear response that you have to the shock. Now, the conditioned stimulus is what? The sound. Very good. So the sound, the tone, is the conditioned stimulus. This is a stimulus that usually is innocuous to the animal, but because of the specific pairing, this is the bell salivation, same kind of pairing that Pavlov first described. the sound now becomes a fearful stimulus. So it becomes a conditioned stimulus. It has been conditioned by this pairing, okay? So these terms are very important for you to know, and of course we went over last time why this paradigm has been so important in our study of fear, and you should know the pathways associated with that we went over last time. So now we want to move on to more global considerations of the study of emotions and also the study of fear, which is psychiatric disorders. It's actually quite stunning when you think about the fact that 1/3 of the US population were symptoms of major psychiatric disorders. That's a huge proportion of the US population. Depression is one of them. It's, this neuropsychiatric syndrome is more prevalent in women. Drug dependency, alcoholism is more frequent in men. I just want to go over a little bit, just a little bit of the history of how we have approached neuropsychiatric disorders in the past. And back in the 16th century, there were specific approaches to heal psychiatric problems. Madness, slowness, mental retardation. They were a little bit blunt, and they basically revolved around, they knew it had to do with the brain. They thought it had to do with the brain. And so their learned approach was to actually release the bad humors from the brain. And to do that, what they did is basically drill a hole in the skull to let anything bad out of the brain that was there. It's called trepanation, and there's a large literature on how this was done early in our history. The early thought was that there were stones in the brain. And while nothing was really removed, all that was removed was the skull. The idea that something was amiss with the brain was common way back in the 16th century. So you might think, oh, well, that's way far back. That's not, we don't have to worry about that. But I also wanted to remind you of this very historic paper that we discussed several weeks ago, the Scoville and Milner paper. Now, I mentioned that, of course, HM, the very famous amnesic patient, was the key focus of that paper, but I also mentioned that this was not just a single case study. This study was focused on 10 subjects, okay? These are the 10 subjects that Scoville operated on. I told you he was a surgeon that was interested in trying to solve neuropsychiatric disorders. He was treating people not only with epilepsy, this is case HM with epilepsy at the top. He was 29 years old at the age of follow-up, 27 years old when his operation was done. But all of these other 9 patients were suffering from things like paranoid schizophrenia, manic depressive psychosis, paranoid schizophrenia, all these schizophrenias. How was Scoville trying to treat them? With bilateral or unilateral resection of the medial temporal lobe. Maybe that would work. Now, can you think about why back in the early 1950s he would think that damage, removing these medial temporal lobe structures would cause any difference, would make any difference for these kinds of emotional deficits? Anything, can you think of any reason why? We've talked about in terms of the history. What was happening before the 1950s, before HM was discovered? What were the ideas there? Yes? Right. Exactly. Everything's concentrated about the brain, but I'm talking about specific hypotheses about what the hippocampus and those medial temporal lobe structures, those limbic structures, might be important for. Yes? They were all about emotions. So what we talked about last time, was that last time? Yes, it was last time, last Wednesday before Thanksgiving, was the early historical ideas that what was called the limbic lobe, the medial surface of the lobe that included the hippocampus, the amygdala, the cingulate cortex, all of these structures were critical, were one, an older part of the brain. An older part of the brain is a part of the brain important for basic functions like fear, like running away from things that are scary, that are dangerous. And so that was a huge theory in the 30s and 40s before HM. So you can imagine Scoville reading these studies and thinking, okay, these areas are important for these kinds of emotional responses. And clearly there's emotional problems in people with manic depression and paranoid schizophrenia. Let's see what this is called psychosurgery. He was doing experimental psychosurgery to see whether removing the hippocampus and or hippocampus and amygdala would cause any improvement in the psychiatric condition. Did it? No, it didn't. And you can see that in several of these patients that were able to be tested, some of them were incomplete because their schizophrenia was too serious to actually be able to have reliable responses on the basic intelligence and memory tests that Brenda Milner was giving. But what they caused, these medial temporal lobe resections, all of these were medial temporal lobe resections, either bilateral, shown here with the B, or unilateral, and he either gave a large medial temporal lobe bilateral resection here or a smaller one. And what they were able to show with with an analysis of all these patients, it didn't help their schizophrenia or their manic depressive disorder at all. But what they found is the larger the extent of the bilateral medial temporal lobe lesion, the more severe their memory impairment. So HM was the best case because he had no neuropsychological disease. He had, he was a normal person with epilepsy, so his tests were much easier to interpret. But overall, they were able to gather and gain information from all of these patients. And of course, all of this surgery stopped after they realized how severe these memory deficits were. So again, just a little bit of a history, but you can kind of trace where these ideas came from. And we can look back and say, oh God, how could they actually do that? But we're probably doing exactly the same thing in areas that we're not very sure about at all. That is part of the scientific process. So from trepanation to psychosurgery, removing medial temporal lobes, we came to an understanding. We're now at the point that we understand that the medial temporal lobe and probably the hippocampus that I'm talking about in the 1950s is important for specifically memory problems, and with the study of emotions, that it's actually the amygdala that seems to be specifically important for processing of fear information. As we talked about at the end of last time, the ability to look at faces in a normal way. When I look at your face, I look at your eyes and your mouth to see whether you're engaged, whether you're bored, whether you're excited. And people with amygdala lesions also have impairments in this basic kind of scanning, face scanning process. Okay, so neuropsychiatric disorders have been a mainstay of movies, and this is part of why your last homework project is the movie report. So we have the story of John Nash, the Nobel Prize-winning mathematician who suffered from paranoid schizophrenia. As good as it gets is somebody with obsessive-compulsive disorder. Away from her, this is more Alzheimer's disease, but Alzheimer's disease associated with dementia. So we want to focus for most of this lecture on, there's so many different neuropsychiatric diseases, but they are very fascinating. There's so much known about them. I want to just focus on schizophrenia. How many people saw A Beautiful Mind? Okay, good. So I thought this was a very nice depiction, a very accurate description of schizophrenia. And you could also look on YouTube for real interviews from John Nash, and it's actually very fascinating to listen to him describe his syndrome and what he's going through during these delusional states. So what does schizophrenia mean? facts about schizophrenia. Schizin means to split, and phren means mind. So schizophrenia refers to a split mind. Having kind of different realities going on at the same time is how early psychiatrists first saw this syndrome. Schizophrenia affects 1 to 2% of the overall population, and it's characterized by what they call dissociative thinking, or impaired logical thought is a key symptom, and that goes back to the definition, split brain. Impaired in logical thought, dissociative thinking. Other very common symptoms in schizophrenia include auditory hallucinations. Some people think that visual hallucinations are part of schizophrenia. It's much more common to have auditory hallucinations. Delusions and changes in affect or emotions are very common in schizophrenia. I should say that visual hallucinations are more common with drugs that induce kind of altered states like LSD. But schizophrenia is auditory hallucinations. And we talked about this is the real John Nash, whose schizophrenia has been controlled with drugs, but is a Nobel Prize winning mathematician. Okay, so I want you to know the difference between positive and negative symptoms in schizophrenia. Positive symptoms refer to abnormal behaviors that are gained over kind of normal behavior. Hallucinations are a positive symptom, mainly auditory in schizophrenia. Delusions, as well as excited motor behavior. The people with schizophrenia get very active and have lots of activity. Negative symptoms are the result of loss of normal function. They can go through slow thought and speech, emotional and social withdrawal. So they say that even in monozygotic monozygotic twins share exactly the same DNA. When one of them is schizophrenic and one of them is not, they say that the one that ultimately develops schizophrenia will be more emotionally withdrawn, more teary, more sensitive as a child, abnormally sensitive as a child. And that could be seen very early on in childhood. Also, blunted affect or emotional expression is common in schizophrenia. One early question was, is this a genetically inherited disorder? And there are two major kind of clues that people have used, that one can use to understand this. And that is to look at twins. The other one is to look at adoption. So let's consider the twin situation first. You can look at monozygotic twins. Those are twins that have the same exact DNA, and dizygotic twins, twins that share half of their DNA. That would be just like your brother and sister. And you could ask whether the twins are what they say concordant for the disease. They both have the disease. Let's say we're talking about monozygotic twins. And if one of the monozygotic twins has the disease and the other does not, it's called discordant. For identical twins, the concordance rate is 50%. That's a huge proportion. So if you have, if you are a monozygotic twin and your other twin has schizophrenia, you have a 50% chance of also developing the disease. This develops in late adolescence, early adulthood. So you could have fewer symptoms early on, although I just said that often you can see early signs of which twin will develop schizophrenia, but you're 50% likely. In dizygotic twins, or just basic siblings, the heritability is 17%. So what does this mean? 50%, you have a 50% likelihood. It means there is a strong genetic component. It's not 100%. Just that would if it was 100% genetic, you would expect 100% of the monozygotic twins to have schizophrenia, but it's 50%. That's still quite high, and suggesting a genetic component, not all dependent on genetics, but a good proportion is. The other kind of study that you can use to try and decide how much of this disease is genetic is studies of adoption. Okay, so two things that we're very interested in is understanding what the genetic component of schizophrenia is, but also what about the environment? What if you grow up in a certain kind of environment? So you can look at adopted children that developed schizophrenia and ask, are their parents more or less likely to have schizophrenia? And you can compare that to a child that is the child raised by his own parents. And so what happens is that if you're adopted and you have schizophrenia, the parents they have a very low chance of actually having schizophrenia themselves. But if you are raised by your own parents, you have a much higher likelihood that your parents also have schizophrenia, either themselves or somewhere in the family. So that suggests that the environment, that is the adopted child, is not being influenced so much by his environment, but it depends more on the genetics of where they come from, where their family history is. If schizophrenia is in the family history, you have a much higher proportion of having schizophrenia yourself. Okay, so two different ways to look at the genetic component: adoption studies and monozygotic twin studies. Okay, so if there is a genetic component, as suggested by these findings, what are the genes that have been identified? And there are a few genes that have been identified as abnormal, but there's no single key gene that you can go and delete and the person doesn't have schizophrenia, or the animal model does not have schizophrenia. But there's all these different factors that we've come to understand over the years. One is paternal age. Paternal age, the age of your father may be a factor. Older fathers are more likely to have a child with schizophrenia. Behavioral aspects, what other kind of clues are there to understand what might be wrong in schizophrenia? And one is eye movement recordings. So you and I, when looking at a dot going back and forth on the screen, can follow that dot very well and very smoothly with our eyes. That's called smooth pursuit eye movement. However, if you have schizophrenia, and this is what the eye tracking looks like, if this is the X direction and this is the Y direction, and this is an accurate representation of where the dot is going. In contrast, this is a patient with schizophrenia. They cannot do smooth pursuit eye movement, and they have these very kind of jagged eye responses, more like little saccades. They can't control their eye movements. Does that remind you of something that we talked about before in other lectures on differences in eye movements? Anybody remember? Yes?
Humans apparently have the best tracking.
Humans have the best tracking, but I'm talking about another disorder that we talked about that is also associated with abnormal eye movements. Yes? Right. Exactly, exactly. So we were talking about, this was from the last lecture on Wednesday before Thanksgiving, we were talking about patients with bilateral damage to the amygdala. From this condition, it causes calcification of the amygdala. It doesn't matter what the syndrome is called, but you have these patients that have just very selective bilateral amygdala lesions. They cannot do fear conditioning, okay? Not surprisingly, given all the data in the rodents. But they also have very abnormal looking patterns, especially when they're looking at faces. So as she was just saying, that normal people will look at the eyes and the mouth. Whenever you look at somebody, you're drawn to the eyes and the mouth to see what kind of expression, what kinds of emotions they are expressing. But this patient with amygdala damage doesn't look at the eyes very much. They're looking at the cheek, they're looking at the ear, they're looking at the tip of the nose. And so that's one of the structures that we've talked about, also associated with kind of emotional not kind of, but strongly associated with emotional processing that shows abnormal eye tracking. Okay, so let's say you were scientists. You had a group of schizophrenic people, and you want to understand the brain basis of schizophrenia. Now you've determined that partially it's genetic, but it's not 100% genetic. You know a little bit about what the behavioral differences are. Certainly the delusions are a major characteristic, but now we know that eye movements are abnormal in these schizophrenic patients. What other questions would you ask to understand the neurobiology of schizophrenia? What would you be curious about based on what we talked about in class before? Yes? Exactly. What specific areas of the brain? That's a fantastic answer. But what else? What else could be abnormal beyond just a particular area? Any idea? Yes? Great. Hormones. Another kind of chemical we've talked about? Neurotransmitters and hormones. Great. Perfect. Those are exactly the answers I were looking for. Brain areas, are they different? What is the brain organization? Are there abnormally large or abnormally small areas in the schizophrenic brain? And two, what about the neurotransmitters or hormonal systems? Are they abnormal? What might be underlying the firing patterns in the schizophrenic brain? Okay. So this was the reminder. We talked about bilateral amygdala lesions impair the use of information. This is just from the last lecture. And controls will look at the eyes and down to the mouth. And here is our subject SM with bilateral amygdala lesions on the nose, a little bit to the eyes, but very abnormal patterns. It's very kind of triangular always. You and I would have triangular looking patterns at faces. Okay, what are the anatomical correlates of schizophrenia? What is different about the schizophrenic brain? Brains of some schizophrenics show structural changes. In particular, the most obvious change when you look at MRI images or get the brains of schizophrenic patients, the actual brains to study, is the size of the cerebral ventricles. The ventricles are enlarged in schizophrenic patients, especially in males, and they remain so after the initial disease onset. When you do a structural MRI, magnetic resonance imaging scan on these people, the more enlarged the ventricles are in the subject, the poorer the response to drug treatment. Okay, so let me just jump here. So here are two sets of twins. Let's say this set of twins here. Which one do you think has schizophrenia? The one on the right or the one on the left? The right. And what about this one? The right. Yeah, that's absolutely true. So it's pretty obvious in identical twins. This means monozygotic twins. But here are more quantitative figures on ventricular enlargement separated out by males and females. As I said here, ventricles are large, especially in males. So here are males with schizophrenia and controls, and we're looking at the age of the subject, and you can see in normal controls your ventricles tend to get a little bit bigger with age. Why? Because there are changes in your brain as you age. There is a decrease in the cerebral kind of volume, and that tends to increase your ventricular size. But at the same age, we're looking at, on the y-axis is the ventricle to brain ratio. And the higher the ratio, the bigger the ventricles. So way down here, from your 20s to 40s, you have a pretty low ventricle to brain ratio. Lots of brain for a little bit of ventricle. But look at the male patients with schizophrenia. Much higher ventricle to brain ratios, very high, and it continues high all through the age, at all ages of patients with schizophrenia. These are the males. Here are the females. Controls, female controls, again, you have lower ventricular volume earlier in life, and it goes up, as I said before. So that part is the same between female and male control subjects. Female patients with schizophrenia, they have a more age-specific increase in ventricular size only in the 30s, but not as much. It goes back down to normal levels through the 40s through the 60s, okay? So everybody see that in males, you have a lifelong increase in ventricular size if you have schizophrenia compared to male controls. But females, it's more time specific. At certain ages, you have increase in ventricular size. Yes? It's hard to say. I mean, that's just, it's not clear whether this part is significant going down, but certainly this part is very significant. So I would say the overall pattern is your ventricles tend to get bigger when you get older. Any other questions about this? So I want you, so now you're starting to kind of make your own graphs on your own for the response time lab, and you'll make graphs for the rat behavior lab. You really need to make sure you understand all different aspects of these graphs. The x-axis, age, different time points of age, the y-axis, ventricular volume. And you should be able to, if given a particular graph with all axes labels, you should be able to tell me what this tells us about differences between males and females, that the response, that the ventricular size is overall and much more consistently larger in males over all age groups, whereas in females it's a much more subtle effect that is most prominent only at a certain age point. Okay, so you should all be able to glean those things about these kinds of graphs. If you're still not clear about different aspects, come and talk to me, and I'll walk you through it a little bit more. Okay, here was our MRI images of the twins. Okay, so let's talk a little bit more about the genes that have been identified. So what helps in these situations are families that get identified that have a very, very high prevalence of a particular disease. And one gene that was identified in a family that had lots of schizophrenia in their history is called DISC1, D-I-S-K-1, SC1. And once you identify a gene in a family that you think might be associated with a disease like schizophrenia, what you can do is put that gene and insert that gene into a mouse model. You do that through what's called transgenics. These are genetic manipulations where you can insert specific genes that you think might be associated with particular syndromes in a rodent and see what it does to his behavior, to his brain structure, and to his brain physiology and anatomy. And so that's exactly what they did. And when they inserted DISC1 into a mouse through transgenic manipulation, what they got was mice with enlarged lateral ventricles. So they knew that this gene, or they suggested this gene has something to do with the structure of the lateral ventricles. And that's a little bit misleading to say. Is it the structure of the lateral ventricles? Do you make ventricles bigger? Ventricles are just holes. What you might be doing is actually making the cortex smaller, okay? And so here's a control mouse with the green are the lateral ventricles on the right and left side, and here's the mutant mice where these green areas become much more enlarged, and that means that that same area that is taken up by the enlarged ventricles is less area for cells of your cerebral cortex and subcortical structures. So abnormal ventricle size size, in abnormal and that is larger ventricular size, centered interest on structures surrounding the ventricles. The structures that surround particularly this ventral part of the lateral ventricles include the hippocampus and the amygdala. And it turns out that there are abnormalities in both of these structures. They tend to be smaller in schizophrenics than in normal controls. It says here, hippocampus and amygdala tend to be smaller. And in particular, they've focused on the hippocampus, or they've identified quite striking anatomical changes in the hippocampus, in the pyramidal cells, the major cell type within the hippocampus. They have a disorganized arrangement. It's not like there's less cells. Well, it's smaller. So there are, the structure is smaller. But in addition, the cells, the pyramidal cells within the hippocampus look disorganized. What do I mean by that? Here is a human hippocampus coming down here. This is anterior, this is posterior. We're taking a slice here. What kind of slice are we looking at down here? Horizontal slice, okay. And so here is the blue hippocampus enlarged here, and we've taken a slice here, and these black dots represent the pyramidal cells of the normal hippocampus. Different sub-areas, you don't need to know the names. This is called the dentate gyrus. Here are the CA fields. And in normal, here are the CA fields, CA2, CA1, of the hippocampus. Again, you don't have to know these names, but normally you just know that these pyramidal cells are organized in a very organized fashion. So here they are depicting just the cell bodies, so you can see the triangular cell bodies, and here you can see the triangle cell bodies with all of the appendages, all the dendrites. Basal dendrites, apical dendrites, but they are all more kind of cleanly organized. However, in the DISC-1 mice, as well as in schizophrenics, where you do histology on the brain, look at the triangles. They're shoved in there every which way. They're not organized where the apical dendrite is always facing the same way. They're facing all other ways. And look at the full cells. You see they're kind of going all over the place. So that's what I mean by disorganized cell layers in area CA1. And the idea here is that during development, these cells are developing in an abnormal way. So you can imagine that it's not just as simple as, oh, one structure, it's just abnormal. It looks abnormal. This may be a developmental kind of abnormality that happens very early in development. Here is actual histology. This is just a drawing. And here is a picture of stained sections where we're looking at a Nissl stain of cell bodies in the CA field of the hippocampus. You can see for yourself that they're more organized in a coherent way, where the apical dendrites, the top going dendrites are all kind of facing the same way, the top of the triangle. But here is the disarray in a patient with schizophrenia. So all the cells are kind of facing in different directions, and the cell layer is not as cleanly organized. It's much more kind of just messy. So you look at this and you start, I hope you start to realize the complexity of this disease. It's not going to be one thing. It's not one disease. Oh, okay, I found one gene and it's going to solve everything. It's going to be lots of complex things that interact in a certain way. And so what I'm trying to give you is an overview in terms of what all these kind of little clues that we have that could underlie eventually and hopefully solve eventually what makes schizophrenia. Okay, what other cortical structural abnormalities are there? There is a thicker corpus callosum. What is the corpus callosum? Yes? Right, the structure, and that is made up of what structure in the cell? White matter. White matter are what part of the cell? Axons. So the corpus callosum is a whole bunch of axons. So what does that mean, that the corpus callosum is thicker? there's more interactions, perhaps, more axons passing from one hemisphere to the other hemisphere. Very interesting. So that means, that suggests connectional differences, kind of major connectional differences in schizophrenic brains. So we have abnormal cell arrangement in schizophrenia, in certain structures, in the hippocampus. We have smaller hippocampi and amygdala. The hippocampus, the smaller hippocampus, has the more disorganized behavior, and we have more, perhaps, interactions between right and left hemisphere than in normals. Some studies show, in addition to all that, some studies show a loss of gray matter. That is, the gray matter, you know the difference between gray matter and white matter, right? What is gray matter made up of? What is gray matter made up of? Cells, cell bodies. So when I say gray matter, I mean cell bodies. And white matter is axons. Good. Okay, so loss of gray matter, loss of cell bodies, loss of the cortical areas in the frontal lobes, and PET studies show less metabolic activity. So let's look at the loss of gray matter first. Here is the rate. This is a representation of the rate of gray matter loss in normal adolescents and adolescents with schizophrenia. The red part means more, significantly more loss during this time. Now, there's some loss in adolescence. Through development, you have lots of cells that develop, and some of them die off. That's normal. But this is normal. Not a whole lot of loss at this time of adolescence, around 1%, in some areas a little bit more. But look at all of this brain loss up here, sensory and motor areas, major brain loss down in the, look at all this in the lateral temporal lobe. These are visual areas, the ventral visual stream processing object information. Look at all that cell loss that's going on in adolescence in schizophrenic patients. This is exactly when schizophrenia is developing. Yes? That's right. That's a good point. So she says, well, how, if gray matter is being lost, how could they have even thicker corpus callosums? Well, maybe it's that the cells that remain continue to have even more interactions with the other side of the brain. We're not positive about that, but that's one implication. Okay, so we talked about loss of gray matter. And what about PET studies show less metabolic activity? The less metabolic activity has been focused on the frontal lobe. What I mean by that is they've shown less metabolic activity in schizophrenics in the frontal lobe compared to controls. And that has led to what's called the hypofrontality hypothesis. Schizophrenia may be caused by underactivation of the frontal lobes. And so here are studies of twins that are discordant for schizophrenia. That means one twin has the disease and the other one does not. The control does not have the disease. Schizophrenia obviously has the disease. On the left is the frontal lobe. These are PET studies. PET stands for positron emission tomography, and it is a measure of activation in the brain. And so we're looking at the frontal lobe on this side, and here's the occipital lobe back here. In controls at rest, you have yellow and red are more activity. So, you know, even at rest, you have mediocre kind of activity in the frontal lobe. And overall in schizophrenia, overall less. Certainly in the right frontal lobe, the greens are much less activity than the yellows. And the back part of the brain looks pretty similar. During a task requiring frontal lobe function, this is where you see the major difference between the twin with schizophrenia and the twin without schizophrenia. This is called a card sorting task. It's a Wisconsin card sorting task, and it's a task that we know is really dependent on the frontal lobe. If you have damage to the frontal lobe, which we'll talk about in a week or two, you have significant deficits on this task. In this task, you're given different cards, and the experimenter plays a mean trick on you. They change the rules on this task without telling you. So the cards have different colors and different shapes, and sometimes the rule is choose all the same shapes. And so you're choosing cards with the same shapes, but all of a sudden, the experimenter will switch the rules, and it will be only choose a certain color. And you'll have to figure out what that rule is by trial and error. And that ability to try different things and use your errors to correct yourself is highly dependent on the frontal lobe. And so you can see in controls, there's lots of activity in the frontal lobe and much less activity in schizophrenia. So this activity, which has been shown in some studies, perhaps not universally, has led to the idea that one of the major problems in schizophrenia is with the frontal lobe and that it's underactivation of the frontal lobe. Now, the second question, so we talked about anatomical changes. This is kind of overall functional activity changes to identify a part of the brain that might be more affected in schizophrenia. Now what we're going to look at neurochemical changes. This has already been mentioned, looking at hormones or neurotransmitters. Hormonal changes have not been studied so much, and I don't think there's drastic changes in hormones, but neurotransmitters have. And in fact, this next kind of part of the story really led to one of the major advances in the treatment of schizophrenia that happened way back in the 50s and 60s. And that happened because of addiction. So people, for many, many years, have become addicted to amphetamines. Amphetamines keep you up. And you can take something like 5 milligrams of amphetamine if you're sleepy and you want to get woken up. If you get addicted to amphetamines, you will end up becoming resistant. To get that same high, that same buzz, you'll need to take something like 5,000 milligrams of amphetamine. So somebody that becomes addicted to amphetamine will be taking these huge amounts of amphetamine. And what has been described in the past is something called amphetamine psychosis. This is somebody that's taking so much amphetamine because he or she has become resistant. They develop what's called psychosis. It's caused by repeated use of amphetamines. And they've noticed, a psychiatrist noticed, that amphetamine psychosis resembles schizophrenia in many important ways, with paranoia, delusions, and auditory hallucinations. So these people that come in, they're addicted to amphetamines. They start looking like schizophrenic patients. What do amphetamines do? Amphetamines promote the release of dopamine. So they stimulate more dopamine release than normal, and they block its reuptake. So it's a double whammy. You're getting more dopamine release, and you're slowing down its reuptake. So the result is much more dopamine around in these patients that are exhibiting amphetamine psychosis. So what helps amphetamine psychosis? Dopamine receptor antagonists, okay? Oh, so just to remind you, dopamine has been coming up over and over again in our discussions. We first started talking about it in the drugs and neurotransmitter lectures. We talked about 2 different dopamine systems, the mesostriatal pathway. Sorry, I think I added this morning just because I wanted to remind you. This came from the lecture that we had. I'll put the newest set of slides with this one included on Blackboard. So we have the mesostriatal pathway to the substantia nigra. What did we talk about that this particular dopamine pathway is so important for? The mesostriatal pathway. What did we talk about? What disease? Yes. Parkinson's disease. Perfect. And what was damaged? What was abnormal in Parkinson's? Yes. Substantia nigra. How was it abnormal? Perfect. It was not producing as much dopamine. So we talked about this mesostriatal pathway, the dopamine projections to the striatum, as causing the tremors, the rigidity, all of the motor problems in Parkinson's disease. Now we're going to focus on the mesolimbic pathway from the ventral tegmental area, mainly to the cortex. And we believe that this is, but also to, I should say, the nucleus accumbens, important for reward, and to the hippocampus. We think that these are the main pathways that might be affected in amphetamine psychosis, as well as perhaps in schizophrenia. Okay, so what they found is they're called neuroleptic or antipsychotic drugs, work by blocking dopamine D2 receptors and can treat both amphetamine psychosis and schizophrenia. Okay, so it was first identified that, hey, dopamine might be a key player here because of amphetamine psychosis, and we knew what amphetamine psychosis did. Because we knew what amphetamine psychosis did in terms of increasing the release of dopamine and blocking its reuptake, obvious response is give dopamine antagonists. It turns out the D2 receptor antagonists were most effective, and added bonus, it not only decreases the psychotic symptoms in amphetamine psychosis, but also in schizophrenia. This has led to the dopamine hypothesis of schizophrenia that states that schizophrenia results from excess synaptic dopamine or increased postsynaptic sensitivity to it. Okay, so and this sounds like a little finding, but it was huge because the discovery of amphetamine psychosis, the focus on dopamine, and the identification of neuroleptic drugs allowed us to have the first effective drug treatment for psychosis, neuroleptic drugs. And the first one that was used very effectively is chlorpromazine. And this is a very common antipsychotic drug, but other drugs that also work on the D2 receptors have been used. As schizophrenic patients go back to their psychiatrist, they may cycle through different drugs. One may work at one point and it becomes ineffective and you have to switch your medications up. But a lot of these are working on D2 dopamine receptors, okay? So think about this for a second. I don't mean to say, aha, now we have the solution. It's one part of the puzzle. These schizophrenic patients still have abnormal development of their hippocampus, abnormal sizes of structures in the temporal lobe, the amygdala and the hippocampus, kind of those structures surrounding the lateral ventricles, and abnormal connections. You're not going to change the abnormal connectivity that may be underlying the larger corpus callosum by giving them chlorpromazine, okay? But the other thing that might be happening is changes in frontal lobe, changes in dopamine function, and so you could treat part of it. It turns out that chlorpromazine, it has been a very effective treatment, and for many years was the only treatment for schizophrenia. Does it work for everybody? No. And that's where some of the problems with the dopamine hypothesis are raised. So what is wrong with the dopamine hypothesis? Well, unfortunately, these same drugs do not work homogeneously or as effectively in every schizophrenic patient. There are patients where chlorpromazine does not work at all. Also, if dopamine is increased, the release of dopamine is increased in schizophrenia, not just in amphetamine-induced psychosis, but in schizophrenia, then you would expect to see higher dopamine metabolites in the schizophrenic cerebral spinal fluid. Do you see that? No. So it's not as simple as the schizophrenics are just spitting out more dopamine normally. Okay, so it doesn't quite jibe with the dopamine hypothesis. The other problem with a simple interpretation of the dopamine hypothesis is that drugs that block the D2 receptors, they block the receptors much faster than symptoms are reduced. So if it's just that you cut off or reduce the activity of D2 receptors, then you should see reduction of these symptoms pretty fast, because we know exactly how effective these drugs are, and the symptoms are reduced much slower. as I said, some patients show no change. So these do not work on all patients. And the kicker is that in exploring other classes of drugs that might work and help ease psychotic symptoms, they found what's called atypical neuroleptics. And these neuroleptics block serotonin receptors as well as D2 receptors. So it may not just be a dopamine story. It could involve serotonin as well. And some of these new drugs, the atypical neuroleptics, actually increase dopamine levels in the frontal lobe and are still effective in some subjects. So this is the same issue that comes up in syndromes like Alzheimer's. We haven't talked about Alzheimer's. But Alzheimer's could be caused by multiple different causes. The end product is the same. You have damage of the hippocampus that causes long-term memory impairments, and that develops into cortical damage that causes the severe dementia, and the damage of the cortex eventually reaches the areas that control breathing, respiration, and heart rate, which will end up killing the subject. Just like in Alzheimer's, where we suspect multiple different reasons for the disease, schizophrenia may be the same. And there may be some schizophrenics where the dopamine hypothesis works. They do have abnormal dopamine, but others that are quite different and that actually improve with increases of levels of dopamine. So again, this is a very, very difficult disease, but hopefully you'll see that all these tools in understanding neuroscience, from the anatomy to the receptors to the neurotransmitters to the behavior, are all working to try and figure out what the heck this is doing. And lastly, I just want to point out a really interesting recent article from Saturday, November 26th, front page of the New York Times, all about schizophrenia. It's called Finding Purpose After Living with Delusion. And I thought it was really fascinating. I put a copy of this on Blackboard, and it talks about one of the few people that actually lives with very severe psychosis, very severe schizophrenia, but has managed to actually live a productive life. This person is a computer programmer, and he's actually been very, very productive and can use his disease state or his particular state of mind in a positive way. Now, it was hard. He was always considered eccentric. And, oh, I'm showing a picture of him. This is him right here. He's also one of the few people that has come out and been very open about his condition. There is a lot of bias against people with schizophrenia. People think that, the general public think that they're likely to be axe murderers, they're likely to commit violent acts. And the truth is, they are not. And in fact, there's a wonderful organization called Bring Change to Mind that helps to dispel these negative beliefs about people with mental disorders. As I said, 1/3 of the US population suffers from some sort of mental neuropsychiatric disorder. And one of the most memorable talks that I ever heard at the Society for Neuroscience meeting was either last year or 2 years ago. And they have this wonderful series called Neuroscience in Society. And they have a non-neuroscientist come and address the whole society. And we're talking about 30,000 people at this meeting. And some of the most memorable speakers have been Christopher Reeve. So when he was alive, he was a big advocate. I think I mentioned that he came to speak. The Dalai Lama came to address the Society for Neuroscience, talking about his interest in trying to understand the neurobiological basis of meditation. And 2 years ago, yes, I think it was about 2 years ago, Glenn Close came. I thought, what is Glenn Close going to say as an actress? But I went because I always like to go when there's always something new that I learn at these talks. And it turns out that she came to talk about this organization that she helped co-found with her sister called Bring Change to Mind. It turns out that she has schizophrenia and manic depressive disorder in her family. And her sister and her sister's son, her nephew, her sister suffers from severe depression, manic depressive disorder, and her nephew suffers from schizophrenia. And they both spoke so eloquently about what it's like to live with these symptoms. And they've both been able to manage their symptoms after long struggles with the right kind of medication. It's not easy to find the right kind of medication because, as I've tried to tell you, it's a very complicated disorder. And so the nephew is an artist, and he is able to live with his condition with drugs. But I thought this article was particularly interesting because of two reasons. One, this person actually has a quite unique kind of disorder. And, well, I hate to call it a disorder, but a unique kind of situation. So for much of his life, he had a syndrome that is common for lots of schizophrenic people. They think they think they're meeting God and Jesus all the time. So they see, somebody with bright blue eyes and think, oh my God, this is, I'm meeting God. And this is part of their delusion. And this was part of this person's delusion. But another part of his delusion was to try and save the world. And so he actually was able to channel this into all of these, he's delusional, he's obsessed with actually trying to improve society. And he does this in a very productive way. So he's very involved in the community, and he's writing letters to his congressman, and he's a bright, intelligent man who's a computer programmer in his real life. But part of his kind of shift that happened when he developed schizophrenia in his late teens was to shift from what he called a very kind of self-absorbed state to a state where, okay, he thought he was meeting God and Jesus, but he also had this severe urge, or this very strong urge, to save the world by doing good for the society. So because of this, he was actually able to parlay this into being able to live with this disease and not only that, the other very amazing and interesting thing about this article is that he is one of a growing number of people suffering from schizophrenia that are starting to come together and talk about their disorders kind of from a first person's standpoint. It's not a psychiatrist trying to understand what this other person is going through, but they're coming together to kind of self-diagnose, but actually try and come up with novel treatments for the disease based on what they themselves, they personally are going through. Of course, these are people whose disease states are managed with drugs. But very interesting, I'll just read a couple of excerpts from this article, talking about what this guy Milt Greek goes through. He says the frightening voices and ominous signs saying that he was damned were no more than embodiments of his very real childhood terror of being cast out as a schoolyard boy's threatened. His search for heaven on earth was in part an attempt to escape that fate, to find a secure place. But it also dramatized a longing to put the world right, a mission that may have started in vain fantasy, as a vain fantasy, but in time became an emotional imperative, a need to commit small acts of kindness, like cooking dinner for a snowed-in neighbor. So one of his ideas is that these psychoses are not just, random things that pop up out of nowhere, but they could also be understood by delving deeper into the history of each individual person. So he sees his delusions that developed as part of his schizophrenia as development from his own personal history. And that is not new, but is actually being treated in a much, much more detailed way by the patients coming together themselves. In fact, he has come up with a new embodiment of a theory. And he says that by listening to the delusions of schizophrenics, he could identify 4 very common storylines. One of them is they believe themselves to be a rescuer on a mission to save a particular group. So Milt himself is on a mission to save the world, and he channels that into all the good acts that he does. The second is a self-loathing person. So there's a lot of self-loathing in some of these delusions, lost in a sense of extreme worthlessness. The third is the visionary, on a journey to spiritual realms to bring back truth. And the fourth is out to transform the world through miracles or contact with deity. So this is his delusions of meeting God and Jesus. So he says these are 4 themes, but many of these themes can be linked back to the personal histories of the person diagnosed with schizophrenia, which is quite new. And very interesting. And in fact, even the psychiatrists are noting that all of the patients coming together to try and give new and more personalized voice to these symptoms are a very, very good thing for the ultimate march towards an effective treatment for schizophrenia. So I would highly recommend that you read this on Blackboard. And any questions about schizophrenia? Okay, so that's all I have for today. And on Wednesday, we will go into cellular mechanisms of plasticity, both in the hippocampus and the amygdala.